Here we're going to talk about nucleic acids and protein synthesis. Let's first, a few definitions here. DNA, deoxyribonucleic acid. This is a nucleic acid that stores the genetic information. DNA is a biopolymer of deoxyribonucleotides, and I'll tell you what those are in a minute. A chromosome is a complex of proteins and DNA, and it's visible during cell division. A gene is a segment of DNA that directs the synthesis of a single polypeptide. DNA is the carrier of genetic information. When a cell is not dividing, the nucleus is occupied by chromatin, and this is a compact tangle of DNA twisted around proteins called histones. During cell division, chromatin organizes itself into chromosomes. Each chromosome contains a different DNA molecule. DNA is duplicated and each new cell receives a copy of the DNA. Each DNA molecule is made up of many genes, segments of DNA that contain the instructions, if you will, to direct the synthesis of a single polypeptide, meaning a protein. Let's talk about a nucleic acid. Again, it's a polymer of nucleotides. So what is a nucleotide? A nucleotide is a 5-carbon sugar that is bonded to a cyclic amine base and a phosphate group. So here we have a sugar, that's our monosaccharide, okay, and it's a 5-carbon sugar. 1, 2, 3, 4, 5 carbons, okay, and notice the ring cyclic structure of the sugar. Here's the phosphate group here, and then this here is a heterocyclic nitrogen base. So there are two types of nucleic acids. We have DNA and RNA, also known as ribonucleic acid. RNA and DNA differ in the sugars. In RNA, the sugar is ribose. In DNA, the sugar is 2-deoxy-D-ribose. Let me see if I have that here. Here are the two sugars. And again, the way we number the carbons here, this is carbon 1, 2, 3, 4, and 5. So this is 2-deoxy-D-ribose, which is present in DNA. And if you notice here, the only difference between D-ribose, which is present in RNA, is the OH group. Okay, so the D-ribose has an OH group on carbon number 2, whereas carbon number 2 in the 2-deoxy-D-ribose only has a hydrogen. So that's the only difference between the two sugars. RNA and DNA differ in their sugars. The bases. RNA and DNA differ in the identity of one of their 4 bases. Thymine is present only in DNA. Uracil is present only in RNA. Adenine, guanine, and cytosine, all 3, are in both RNA and DNA. Let's take a look at the heterocyclic amines that are found in nucleic acids. Again, as I said, thymine is present only in DNA. You will not find thymine in RNA. Again, like everything else in chemistry, though, there could be some rare exceptions. Uracil is only present in RNA, so you will not find uracil in DNA. Adenine, guanine, and cytosine are present in both DNA and RNA. So here are the bases. Purine is the parent, so adenine and guanine are the purine bases. And again, we find adenine and guanine in both DNA and RNA. Here we have the pyrimidine bases, and the pyrimidine bases are cytosine, thymine, and uracil. Cytosine is found in both DNA and RNA, and thymine only in DNA, and uracil only in RNA. Thymine can occur in a few cases of RNA, but again, it's rare. Okay, for our purposes, we will just assume that it does not. Now, we've already defined what a nucleotide is. Now let's define a nucleoside. A nucleoside is a 5-carbon sugar bonded to a cyclic amine base. So it's similar to a nucleotide, but it just does not have the phosphate group. Okay? Nucleosides are named with the base name modified by the ending osine, osine, for the purine bases, and idine for the pyrimidine bases. So you have to make sure you know which are the purine bases and which are the pyrimidine bases. For example, here we have ribose and adenine to form the nucleoside adenosine. So a nucleoside plus a phosphate, of course, equals a nucleotide. Each nucleotide is what we call a 5' monophosphate ester of a nucleoside. Nucleotides are monomers of DNA and RNA polymers, okay? So you can think of RNA and DNA as being biopolymers, okay, that are made up of nucleotides. Now, how do we name a nucleotide? Deoxy is added to deoxyribose nucleosides. Numbers with primes are used for atoms in the sugar. Nucleotides are named by adding 5' monophosphate at the end of the name of the nucleoside. Okay, so for example, here we have deoxyribonucleoside. Okay, and anyway, we add a phosphate to that, and now it becomes a deoxyribonucleotide. Now, we didn't talk about phosphates in the previous chapter, but basically, if you look at this, all right, this here is very similar to an ester group, but instead of a carbon here, we have phosphorus. So we would call this a monophosphate ester bond, okay, or a phosphate ester bond, okay? So same group as an ester, except there's a phosphorus atom in place of the carbon. In this case, we call this a 5' monophosphate ester bond. Why? Because oxygen here is attached to carbon number 5 of the sugar. Remember, 1, 2, 3, 4, 5. So we have a 5' monophosphate ester bond. Adenosine 5' monophosphate, and we abbreviate that AMP, and deoxycytidine 5' monophosphate. And this is abbreviated CMP with a small d in front of it to show that it derived from the deoxy sugar. So nucleotides that contain ribose are classified as nucleotides, and those that contain 2-deoxy-D-ribose are known as deoxyribonucleotides. And we designate them by leading their abbreviations with a lowercase d. So right here, we have adenosine 5' monophosphate. We're going to see mainly abbreviations here, AMP. So this is a ribonucleotide, and our deoxyribonucleotide, in this case we have deoxycytidine 5' monophosphate, and this has that lowercase d in front of it. Let's take a look now at the phosphate groups. And phosphate groups can be added to nucleotides to form either diphosphates or triphosphate esters. So in this case here we have a monophosphate ester, here we have a diphosphate ester, and here we have a triphosphate. And this molecule here is very important, adenosine triphosphate, ATP. And this is our high energy molecule. And basically the energy is released when a phosphoryl group is transferred from the ATP to another molecule. So it's the hydrolysis reaction that actually results in the release of free energy, if you will. Remember, free energy is available to do work. Okay, and we're going to talk about coupling biochemical reactions a little bit later. Here we have the bases, adenine, guanine, cytosine, thymine in DNA, and then the bases that we find in RNA. And here, the deoxyribonucleosides would be called deoxyadenosine, deoxyguanosine, deoxycytidine, and deoxythymidine. And then it also shows the naming of the deoxyribonucleotides, okay, using these bases. I'm not going to tell you to memorize these, but you need to know what I'm referring to when I talk about these. So take a look at this table. So let's go ahead and take a look at the structure of nucleic acids. Again, remember, nucleic acids are the polymers of nucleotides. So they're biopolymers. Just like proteins are biopolymers, the monomers are the amino acids. Well, the nucleotides are connected in DNA and RNA by phosphate diester linkages between the group on the sugar ring of one nucleotide and the phosphate group on the next nucleotide. Let's take a look at this. Here, in general, a nucleotide chain will have a free phosphate group on carbon number 5, okay, which it does here at one end, and that's known as the 5' end, and a free OH group on a 3' carbon at the other end, the 3' end. So this shows linkage. This is a dinucleotide. Here we have a linkage here between the oxygen on the phosphorus and the 3' carbon, the one sugar, and here a linkage here between another oxygen on the phosphorus and the 5' carbon on the other sugar. We call this a phosphate diester link. So that is a phosphate diester link. We have 3 nucleotides. We have a trinucleotide, okay, which is made up of 3 of these linkages. So what we do is when we read a nucleotide sequence, we read it starting at the 5' end. We identify the bases in order of occurrence. And we only use one-letter abbreviations of the bases. So we use a capital A for adenine, a capital G for guanine, a capital C for cytosine, capital T for thymine, and a capital U for uracil in RNA. So we would represent this nucleotide here as T for thymine, A for adenine, and G for guanine. So you can write these with a hyphen in between letters, or just go ahead and write the letters. So either way is good representation. It just basically is going to depend on the text, okay? So you can use hyphens between the letters, or you can eliminate the hyphens. Again, it's just going to depend on your text. And notice we have these are linked together by the diphosphate ester linkages. And also notice that we have a free phosphate group on the 5 position, and then we have on the 3 position that OH group. Okay, the structure and function of a nucleic acid will depend on the sequence in which the individual nucleotides are connected. Okay, just like in proteins. Okay, remember the function of a protein is going to depend on the sequence or in the order in which the individual amino acids are connected. So the differences between the different nucleic acids are again provided by the order of the groups that are bonded to the backbone and the bases. In a nucleic acid, the backbone is the sugar and phosphate, and similar to proteins, there's no variation in the backbone, right? Because you have those alternating alpha carbons and peptide links. Well, same with nucleic acids. What you have is a sugar-phosphate backbone. And you can think of the bases as being similar to the side chains of the amino acids in the protein. Let's take a look here at the structure of the DNA. Basically, the structure of DNA came from X-ray diffraction studies. Okay, probably a lot of you don't know what that is. But anyway, James D. Watson and Francis H. Crick, they were working at Cambridge University, and they designed and built a scale model of a DNA molecule. Anyway, the structure of DNA, according to Watson and Crick, consists of 2 polymeric strands of nucleotides in the form of a double helix, with both nucleotide strands coiled around the same axis. And here, I'll show you this figure. So you can think of both of them coiled around the same axis. Anyway, along each strand are alternate phosphate and deoxyribose units with one of the 4 bases, adenine, guanine, cytosine, or thymine, attached to deoxyribose as a side group. The double helix is held together. Let me get to this slide here. The double helix is held together by hydrogen bonds extending from the base on one strand of the double helix to a complementary base on the other strand. The 4 bases are flat in their ring structures. So you can think of the DNA structure as that of a ladder that is twisted into a double helix. So here's our ladder. Here are the bases. This corresponds to the backbone of the 2 strands. And we see that the bases strands are held together by hydrogen bonds between bases. And we'll talk about that in a second. So, you can think of the rungs of the ladder being kept perpendicular, okay, to the twisted railing. The phosphate and deoxyribose units alternate along the 2 railings of the ladder, and 2 nitrogen bases form each rung of the ladder. So in the Watson-Crick model of DNA, 2 polynucleotide strands fit together best when a purine base is adjacent to a pyrimidine base. So this allows for 4 possible base pairings. So A compare with T, A compare with C, G compare with C, and G with T. Only the adenine, thymine, and guanine cytosine base pairs are able to hydrogen bond together. So under normal conditions, an adenine on one polynucleotide strand is paired with a thymine on the other strand. And of course, a guanine is paired with a cytosine. This pairing results in A to T and G C ratios of 1:1. So the hydrogen bonding of complementary base pairs is shown in the following figure. So again, thymine will hydrogen bond with an adenine and guanine with cytosine, but we'll never find a guanine for example, hydrogen bonding with an adenine. And there's a reason why. Look at the base pairing here. Thymine and adenine pair, and they pair together through these two hydrogen bonds. Okay, so there's an oxygen atom here, and there's a hydrogen bond between an oxygen atom here and a hydrogen atom that's bonded to a nitrogen on the other base, and the same here. Notice that cytosine and guanine, there are 1, 2, 3 hydrogen bonding. It wouldn't be possible for an adenine to bond with a cytosine or a thymine to bond with a guanine. So this is how the base pairing works. So an A base is always opposite a T in the other strand, and a C base is always opposite a G. Let's go ahead and take a look here at this problem, and the problem states what sequence of bases on one DNA strand is complementary to the following sequence on another strand. So here we're looking at this here. So, the key is, if the sequence of one strand is known, then we can determine the sequence of the other strand. We'll discuss in a little bit that a cell is able to chemically read one strand in order to synthesize its chemical partner. The double helix is an important part of the DNA structure. However, it does not explain how the very large DNA can be packed into a cell or the even smaller cell nucleus. For example, a human DNA molecule can be extended almost 10 centimeters in length and yet is contained in a nucleus with a diameter that's about 100,000 times smaller. To begin the necessary packing, the DNA is looped about small aggregates of positively charged histone proteins. Hundreds of these aggregates are associated with each DNA molecule, so the DNA is foreshortened and has the appearance of a string of pearls. Further condensation is achieved by wrapping this structure into a tight coil called a solenoid. Finally, the solenoid nucleoprotein complex is wound around a protein scaffold within the nucleus. By following this complete procedure, human DNA can be packaged into the nucleus as condensed chromosomes. It's like a high-density computer disk. That DNA takes up only a little space relative to the large amount of genetic information it contains. So let's go ahead here. What sequence would be complementary to this sequence here? So what we would have is we have a G with a C with a G, C, G, T, A, T, G, C, T, A. And remember, the complementary, the first strand is from the 5' to 3'. The complementary would be the 3' to the 5'. Yeah, here's another representation of the DNA double helix. Again, this represents the backbone. Here we see one, it's a little difficult to tell, but we see the bases in blue, and the sugar phosphate backbone, and so on. Okay, so there are 3 processes that are involved in duplication, transfer, and use of genetic information. First is replication, and this is the process by which a replica or identical copy of DNA is made when a cell divides. This is basically DNA synthesis. Transcription, this is the process in which DNA is used to synthesize RNA. This is messenger RNA. Okay, we'll talk about that later. And then, of course, there's translation, the process by which the genetic messages carried by RNA are decoded and used to synthesize proteins. This is protein synthesis. So let's talk a little bit about replication. What we have here is we have a strand of DNA. Replication is one of the most complicated enzyme catalysis processes in life. There are enzymes that are required to unwind the DNA before replication and to repackage the DNA after synthesis. The 2 template strands are copied differently. One daughter strand goes directly toward the point at which the templates are unwinding, while the other daughter strand is synthesized away from this point. In this way, the same enzyme catalyzed reaction can be used to synthesize both strands. The one daughter strand that has been created in small fragments must be connected before replication is complete. So while one DNA strand is formed by continuous synthesis, the other new strand is formed by a repetition of fragment synthesis followed by a coupling reaction. Here we have DNA, okay, a helix, which unwinds. And notice here, here a strand continuously grows, and that's from the 5' to 3' direction. Replication proceeds in the 3' to 5' direction on both strands, okay? So one is being replicated here in a continuous growing strand, and the other one is the lagging strand, we call it. It replicates in fragments, okay? It's the only way, because as we said, replication proceeds in the 3' to 5' direction on both strands. This one here, again, is replicated in fragments, okay? And then there's special enzymes that are needed in order to join these fragments together. Anyway, when strands are complementary, only one new strand can begin at the 3' end of the template strand and grow continuously as the point of replication, or the replication fork, okay, moves along the original DNA. And again, the other strand's growing in the opposite direction, but it does not grow continuously. It's a very, very, very complex replication. It's very complex with all the enzymes. Anyway, I'm not going to have you memorize all of the enzymes that are involved or anything like that. When you think about it, you consider the magnitude of the job in the replication. The total number of base pairs in a human cell is about 3 billion base pairs. And a single DNA chain can contain up to 250 million base pairs. And yet, the base sequence of these huge molecules is faithfully copied during replication. And yeah, do errors occur? Sure, a random error will occur, maybe about once in each 10 to 100 billion cases. The complete copying process in human cells takes about a few hours, several hours. It's, again, a very, very complex process. Each new DNA molecule then is going to contain a strand from the original DNA plus the new strand. Let's talk a little bit about the structure and function of RNA. RNA molecules are smaller than DNA molecules. They're structurally similar. Both are sugar phosphate polymers and both have heterocyclic bases attached, but there are some differences. They differ in composition. The sugar in RNA is ribose, and in DNA it's deoxyribose. And remember, uracil, the base, is present in RNA instead of thymine, which is present in DNA. They do differ in size and somewhat in structure. RNA molecules are much smaller, okay, than DNA molecules. And even though RNA may be single-stranded or double-stranded, it will not form a double helix. DNA is only has one function, and that's to store genetic information. RNA has different functions. So there are 3 types of RNA. We have ribosomal RNA, and this is complexed with proteins in the ribosomes. All right. This is outside the nucleus, but within the cytoplasm of the cell. And this is where protein synthesis takes place. And again, each ribosome is a complex where it consists of about 60% ribosomal RNA and 40% protein. Okay, and usually has a molecular weight of about 5,000 atomic mass units. So that's pretty high. We then have messenger RNAs. Okay, and messenger RNAs are the nucleic acids that carry the information that's transcribed from DNA in the cell nucleus out of the nucleus to where the proteins will be synthesized. And then, of course, we have the transfer RNA. And transfer RNAs are smaller RNAs that deliver amino acids one by one to the protein chains that grow at the ribosomes. So let's talk a little bit then about transcription, RNA synthesis. RNA is synthesized in the cell nucleus, and there are different enzymes involved than there are with DNA replication. RNA is synthesized as a single strand. So only one of the DNA strands will be transcribed, and that will be the template strand. Its complement in the original helix is the informational strand. This is the strand that is not used for RNA synthesis. So RNA is a copy of the informational strand, okay? And you have a thymine replaced with a uracil. Messenger RNA is a duplicate of the DNA informational strand, and again, T is replaced with a uracil. The RNA strand grows from the 5' to the 3' direction. So let's take a look at this. In the transcription process here, it begins when RNA polymerase recognizes a control segment in the DNA that precedes the nucleotides to be transcribed. The genetic code relies on triplets of consecutive bases. Basically, we call those codons. We'll talk about those in a minute. The triplets carried by messenger RNA code for amino acids that are then assembled into the proteins. Okay? So what happens is the RNA polymerase moves down the DNA segment that's to be transcribed, and it adds complementary nucleotides one by one to the growing RNA strand. Okay? Transcription will end when the RNA polymerase reaches a nucleotide sequence that actually signals the end of that particular sequence to be copied. Okay? So at the end of a transcription, the RNA molecule will contain a base for every base that was on the informational DNA strand from the start to the top codon. The messenger RNA is not ready for protein synthesis yet. It turns out that the genes will only copy about 10% of base pairs in DNA. A gene will begin in one small section of DNA, which is called an exon. Okay, an exon is a nucleotide sequence in a gene that codes for part of a protein. But anyway, a gene will begin in one small section in the exon, be interrupted by a section called an intron. An intron is a nucleotide sequence in messenger RNA that does not code for a part of a protein. The introns are actually removed before the messenger RNA proceeds to protein synthesis. Okay? Basically, they're taken out, and then the remaining pieces are spliced together. Possible functions of introns, or non-coding DNA as we call it, still under study. There has really been no clear-cut function assigned to the introns. Okay? Some possibilities, maybe they're leftovers from earlier stages in evolution. Okay? But that's another time, that's for another time, another place. The genetic code. Actually, this table is not in your book. Let's go ahead and talk about a codon. Okay? And a codon is a sequence of 3 ribonucleotides in messenger RNA chain that will code for a specific amino acid. It also includes a stop codon to stop translation. The genetic code is the sequence of nucleotides coded in triplets, codons, in messenger RNA that determines how the amino acids will be connected in the protein. Codons are written in the 5' to 3' direction. Basically, you have your 20 amino acids, right? Most amino acids are going to be specified by more than one of these codons. And I'll show you the relationship between the DNA informational strand, the template strand, the messenger RNA, and then how the protein is synthesized. Here it says this is the DNA informational strand. What is the template strand and the messenger RNA for the following? Okay, so this is our informational strand, and we have the sequence here. So now we want to determine the DNA template strand. So here's the DNA template strand. Again, we have to go from the 3' to 5'. So here we pair up a T with an A, T, C, G. Okay, G, C, G, C, T, A. C, G, A, T, T, A, C, G, C, G, G, C, G, C, T, A, G, C. Okay, A, T, C, G. Okay, so we're able to write the template strand from the informational strand. Now, the messenger RNA, remember, is taken from the informational strand. Okay, so basically, the messenger RNA is the informational strand, but each thymine is replaced by a uracil. Okay, So it is the informational strand, but we replace each thymine with a uracil. So that's fairly easy. And then as far as the protein goes, well, if you go to the table in your book, what you do is you go ahead. Here we have our first codon. Okay, we have an AUG, and the AUG corresponds to methionine amino acid. CCA corresponds to proline. GUA corresponds to valine. GGC to glycine. CAC to histidine. UUG to leucine, and UCA to serine. So this would be the amino acid sequence for this particular messenger RNA. Disregard this page number here. So remember, we have the DNA informational strand. Is this the same as this one? No. Here's a DNA informational strand. So you have the informational strand is read in the 5 to 3' direction. Here's our informational strand. The complement of that is the template strand. This is the strand that is transcribed. So the messenger RNA is actually the complement of this strand, with each adenine replaced by a uracil. Or, remember, the messenger RNA is the DNA informational strand, same sequence, except that each thymine is replaced by a uracil. Let's go ahead then and talk about the structure of transfer RNA. Transfer RNA has this cloverleaf type structure, and what it contains is an anticodon triplet right here and a covalently bonded amino acid at its one end. The anticodon of each transfer RNA is complementary to a messenger RNA codon, always the one designating the particular amino acid that the transfer RNA carries. Every cell contains 20 or more different transfer RNAs, each designed to carry a specific amino acid, even though they're all similar in overall structure. We can see it's a single polynucleotide chain. It has kind of a partial helical structure that looks like the cloverleaf. Okay? So each amino acid is bonded by an ester linkage between the carboxyl group of the amino acid and the OH group of a ribose at the end of the 3' end of the transfer RNA strand. Okay, so that's at this end, right? And then, of course, here we have our anticodons. So the transfer RNA carrying the amino acid, let's say it was carrying leucine, for example, which is coded for by CUG in the messenger RNA, has the complementary sequence GAC as its anticodon. So this is how the genetic message of nucleotide triplets, or codons, is translated into the sequence of amino acids in the protein. When the transfer RNA codon pairs off with its complementary messenger RNA codon, then the amino acid is delivered to its proper place in the growing protein chain. Yeah, I just explained everything on this particular slide, and I used the leucine as an example. Okay, good. All right, so translation. This is transfer RNA and protein synthesis. So again, synthesis begins at the ribosomes outside of the nucleus, but still within the cell cytoplasm. So the messenger RNA connects with the ribosome, and the amino acids attached to transfer RNA are delivered one by one to the growing protein chain. So what we have here is we have initiation. So the protein synthesis begins when a messenger RNA, the first transfer RNA, and a small subunit of a ribosome come together. So the first codon on the end of a messenger RNA and an AUG codon acts as a start signal for the translation machinery and codes for a methionine carrying transfer RNA. The initiation is complete when the large ribosomal subunit joins the small one, and the methionine bearing transfer RNA occupies one of the two binding sites on the united ribosome. The three stages are initiation, elongation, and termination. We already talked about the initiation, okay? Elongation, this is this step here. All right, so we have the initiation, now the elongation. Next to the first site on the ribosome is going to be a second binding site where the next codon on the messenger RNA is exposed, and the transfer RNA that carries the next amino acid will be attached. Remember, we have all these transfer RNA molecules in the vicinity, okay, so they can all approach, but only one with the appropriate anticodon sequence is going to bind. So once the second amino acid has arrived, what we have is an enzyme in the large subunits that catalyzes formation of a new peptide bond. And basically what happens is the bond of the amino acid 1 is broken with its transfer RNA. The first transfer RNA is then going to leave the ribosome, and the entire ribosome then is going to shift 3 positions. Remember, that's going to correspond to a codon along the messenger RNA chain, and this is a process that we'll call translocation. So basically, now we have a second binding site that is opened up, and it's available now to accept the transfer RNA that carries the next amino acids. So these 3 elongation steps will now repeat. Okay, so the next transfer RNA, or the appropriate one anyway, will bind to the ribosome. A peptide bond will form. It attaches a newly arrived amino acid to the growing protein chain, and then translocation takes place in order to free up a second site for the next transfer RNA. So then, finally, our last step is termination. When the protein synthesis is complete, a stop codon, it's usually a UAA, a UGA, or a UAG, will signal the end of the translation process. Basically, there's an enzyme-catalyzed reaction that cleaves the polypeptide chain from that last transfer RNA, and then the messenger RNA molecules and the transfer RNA molecules are then released from the ribosome, and then the 2 ribosome subunits will then separate. This is just a very basic overview of the replication, transcription, and translation processes. So here's a problem for you. What tetrapeptide would be synthesized from the informational DNA sequence given? Okay, so this is the informational sequence. So let's go ahead and write the template strand, okay? And so that would be 3', right? A C will pair with a G, a T with an A G with a C, an A with a T, a C with a G with a C with a G, and a G with a C. Here we have a C, a thymine, and an adenine, thymine and adenine, and 5'. So this is the template strand. Now let's take a look. I'm looking for the table in the book here. Oh, here it is. Okay. So here's what we have. Now let's do the messenger RNA sequence. Okay, and there are 2 ways we can do this. If we are given the informational strand, then we know that the messenger RNA strand is simply the informational strand sequence, but each thymine is going to be replaced by a uracil. Okay? Or if we were only given the template strand, then the messenger RNA is the complement of that strand, and again, for each adenine, we would replace that with a uracil, not a thymine, because we don't find thymine in RNA. Since I have the informational strand, this is just going to be C, C, and then T is going to become a uracil. And that's going to be G, A, C, G, C, G, and G, and now both of these T's will be replaced by uracils. So that's our messenger RNA. Now, from this, now I can go to the table in the book and I can determine the protein sequence. So let's see. For CCU, that's going to be a proline. Okay. And then I have GAC, so I looked that up. G, A, and C. I have an ASP. And then for GCG, let's see, G, C, and G, that's going to be an alanine. Okay. And then for GUU, I'm going to have GUU. That's going to be a valine. Okay? If I'm given the informational strand, I can determine the messenger RNA from that. It's simply the informational strand with each thymine replaced by a uracil. But if I was given the template strand, that would be the complement of the informational strand, and each thymine would become a uracil. If you have any questions, please let me know.